this is now possible. And this is really hot off the press over the last few weeks that we've gotten some of the in vivo data, which is really kind of the metric that people look at to say, hey, this is cool. Cancer's been cured in petri dishes millions, maybe billions of times. So the bar is a little higher than that, but we're starting to show that this is possible.
Welcome to AI and Healthcare. I'm Mika Newton, your host, and I am super excited to have you here for this interview series. We're going to be discussing healthcare and how AI is impacting that. I am really excited today to have Tom Neyarapally, who I've known for over 10 years and has been both a friend and a colleague in this industry with me for a long time. Tom was a founding member at Atea, Semaphore, and now at Archetype Therapeutics, where he's held a wide variety of roles across corporate development, R&D, commercial, and now as founder and CEO. So Tom, thank you so much for coming on this series with us. Really excited to have you here.
Thanks, Mika.
So look, both you and I have been working in healthcare and AI for a long time. And I think we've been thinking about AI probably, I would say collectively between us for probably more than double the amount of time that we've known each other. Why is it that we look at technologies like this? And I'm thinking back to Atea, which I know is, GNS, and the development of technology and algorithms. What has been the issue in drug discovery, especially in oncology? And why is it still taking us so long, do you think, to bring new innovations to the market?
Yeah, it is funny because we were having these types of conversations 10 plus years ago, thinking that we were right on the cusp of making everything happen to really make big changes and make big discoveries in drug discovery using these types of tools. And I think ultimately it comes down to a combination of things that needed to come together and that have now come together in a truly transformative way. One is having the right data. And so we now, through all the latest and greatest innovations that have occurred in molecular profiling, as well as in the area of collecting clinical data from patients, both of those elements, at least for what we do in my current company, are critical, and I think are critical in general in AI for drug discovery, that we now have access to thanks to a myriad set of companies that work on different aspects of those data problems. So the data is part of it. Having the right AI and ML tools, and that, as you know, continues to evolve over time. Every month, there are new great tools that become available and that everyone kind of stands on the shoulders of giants and continues to build on these AI and ML tools. More recently, it's been things such as generative AI, but it's really been this continuum over the last 20 years of machine learning and now what we call AI tools that have increasingly become available to the average user to be able to build from. And then the right compute infrastructure. Not everyone had access to hundreds of thousands of processors if they wanted to 10, 20 years ago. I remember using these physically massive machines, IBM supercomputers, that now, we have that kind of power accessible to millions of people through AWS and Google and other types of services. So it's really a combination of all of those things. And then, of course, the old school stuff is you still need the clinical and scientific expertise to be able to leverage those other three capabilities. But I think those first three things that I mentioned have now come together in a meaningful way that is materially different than where we were 10 years ago.
Yeah, it's amazing. As you said that, I was remembering back to, again, probably 20 years ago, where I sold silicon graphics workstations, right, and machines. And I remember the very first kind of, compute farms that we were building. We were building servers together. We were multi-processor, and I think 16 processors, right, not thousands of processors, was like the state of the art. Can you think back like a little bit over your experience? And I appreciate what you're saying about how these things change. Like, I think many people feel like AI, the generative AI stuff now is very exciting. And we'll talk about that a little later. But in the older machine learning kind of algorithm development stuff, do you remember some like specific things that really addressed like time? Like, what was the impact of kind of like the before and after around some of the machine learning algorithms and things that happened? Because I feel like now we're at the stage where we're taking that to the next generation. But I also think we have to look at what did we really get out of the older generation of these tools?
Yeah, if I think back to, you were mentioning some of my prior experience, going back almost 20 years to working with what's now called Atea, what used to be called GNS Healthcare. When I first met Colin Hill and Aya Khalil, who were the original co-founders of that company, going back 25 years when I met them in grad school at Cornell, they were leveraging, as I mentioned, these physically massive IBM supercomputers. They were called Blue Gene machines. And a lot of the innovation there was just being able to tailor the machine learning algorithms to take advantage of these architectures that emerged, the computational architectures that emerged. So the first set of innovation was just, how do I actually take advantage of the fact that I can use thousands of processors at once? So there was all sorts of ML algorithms about how to spread out the work. And then the fact that that was possible allowed us in that company to start looking at learning ensembles of models. So imagine you're trying to build a model of a particular type of cancer, let's say colon cancer, and you're trying to understand all the different nodes, let's say the expression of a set of genes in a network. And you're trying to get to an answer that's under constrained. There's not enough data to learn the model that explains all of that data. So we started to take advantage of these architectures where you might have access to 1,000 processors, and you would say, let's learn 1,000 models. Each model will evolve on its own processor, and we will then use the ensemble of models together to ask a question. What happens if I knock down gene X? Becomes a question that we ask not one model, but 1,000 models, because we've innovated and done the software engineering to be able to take advantage of those types of architectures. So those types of things weren't possible when we didn't have access to those types of compute architectures. And then once those started to become possible, to me, that was the first wave of innovation, was just how do you take advantage of the fact that there's just fundamentally different types of computing available to us, which now seems like old hat, of course, with everything in the cloud and so on, but really wasn't back then 15, 20 years ago. And then it became a game of, okay, now that we can do that, how do we squeeze more juice by getting better and better algorithms to be able to take advantage of these architectures? And that was how that particular company evolved over time. But it started with that ability to take advantage of a large number of processors versus one processor at a time.
Yeah. So like to your point now, we've got this amazing computational capability, right? We have algorithms that have actually been tested and gotten, in some ways, far more sophisticated than I even expected them to get in a pretty short time period. So when we think about kind of drug discovery and the time it takes and the cost, I mean, we're still talking about billions of dollars over a period of like a decade or more to get something from, let's call it target identification, early stage lead gen, all the way through clinical trials and actually, marketing approval and commercial launch, and then all of the like moving from an indication to another. Where do you think the big opportunities in shrinking the time scale are? If we look at kind of all the steps of drug discovery, going from that early target validation, lead identification, lead optimization, right? Initial, preclinical work, clinical work, phase 1, 2, 3 studies, et cetera. Like where's the big opportunity in your mind to kind of shorten the time to market?
Yeah, there are really multiple opportunities within that wide continuum that you just described. In the world that I'm working in right now, the ability to connect different parts of what that linear progression that you just described, I think, is really important. So in what you described, someone in a lab, perhaps in an academic environment, discovers a new piece of biology, and then they publish it. And then a bunch of people, both in pharma companies and academia and elsewhere, try to go characterize that particular, for example, drug target. It might be a protein. And so there are structural biology companies like Proteros, for example, that do that type of work and have been doing that for a long time and are masters of that. So, freeze the protein, do cryo electron microscopy to characterize it, and then, or do X-ray crystallography to characterize it. And then after that, go try to find molecules that bind that target. And there might be different kinds of physical high throughput screening approaches that take months and a lot of money and time to do. And then you try to find a hit, and then you go on down the cycle that you were describing through the various preclinical models. So make sure that it works in a petri dish, make sure that it works in animals, and then get into the clinic, and then those series of phases of clinical development that occur before the drug is approved. That process, 10 years, maybe longer. So part of the problem is that some of those pieces that I just described are disjointed. So once that target got discovered in an academic lab somewhere, it gets thrown over to that next group of people I described, and they work on it kind of out of the context of a set of patients. And so ultimately you find a drug through that long process of many years, then you go try to test it in real people. And to me, that's one of the disconnects, because at that point, you're pretty far divorced from that original exploration of the biology. And now you're saying, hey, does this actually work in a group of actual people that have this disease? And these diseases are not monoliths. So not all lung cancer is the same. Not all prostate cancer is the same. Patients are defined by the therapies that they've had before, by their genetic background, by the genetic background and the other omic background of the tumor, for example, if we're talking about cancer. And so all of these other things can get lost from that process of trying to get a drug to hit the target. And so some of the solutions that are coming through now, including some of the stuff that we're doing at Archetype, is to not lose that patient-centric focus and, in fact, start with the patient, characterize a set of patients that have a disease, and try to find drugs and screen at massive scale for drugs that will move the needle for that particular patient cohort, that will move the biology of that set of patients from illness toward wellness. And so that's a fundamentally different way to think about drug discovery. And as we're proving through some of the work that we've done that we'll talk about later, that seems to be working. And so we can short circuit a lot of the failure that's built into that process because of the disjointedness of the pieces that we described.
You know, as you were talking, it reminds me about a project that I followed pretty closely in a group out of Eli Lilly and Company that was called the Chorus Group. I don't know if you ever ran across them, but what always struck me about them is they had this idea, it just reminds you what you were talking about, that for hard drugs to develop, for things that the risk of failure was going to be high, or which is a lot of drug discovery, especially in oncology or rare diseases, et cetera, it made sense to kind of either, and I think in their case, they were mostly like leaving out kind of the standard parts of the process and focusing almost exclusively on this idea of proof of concept. Like if I could show that I could get from my clinical kind of academic idea to something that worked in a human being, in a patient, as quickly as possible without worrying about necessarily all of everything that had to happen right on a regulatory perspective, then I could go and spend all the money and time and effort to do all the, kind of heavy lifting along the way. But I would be spending that money knowing that I had a much higher likelihood of success. So I could reduce the risk. So kind of what I heard you saying in that is now that these computational methods, right, are allowing, and they were just reorganizing the structure of the project structure itself, but that we could do that kind of in a parallel way, really in silico. Am I understanding kind of where you were going, right? We could keep that concept there?
Yeah, exactly. I mean, the idea is rather than starting in kind of some esoteric biological discovery process, let's start with a set of patients that we're trying to solve a problem for. Let's characterize those patients clinicogenomically. And again, through the work of many different types of companies, that's now possible. The diagnostic companies like XCure's, your company, other types of companies, were able to get access to that patient data to do that. So that wasn't possible 20 years ago. It's now very possible to characterize a set of patients, say, these patients have unmet need. We can get their clinical data, we can get their genomic data, and we can build classifiers that predict how those patients are going to fare, and also describe the biological states of patients that fare well and fare poorly. And then we can say, let's start with a goal to move that set of patients from states associated with illness to states associated with wellness. So we can do that computationally. Yes, we can describe the patients, which we've been able to do for 20 years and with increasingly better data, but now we can actually act on that information using these computational approaches and say, let's find drugs that will push those buttons, move the needle for those patients specifically. And that's not to say that all the kind of canonical biology and so on gets thrown out the window. That's absolutely important, and you need that scientific and clinical expertise to validate all this stuff. But to your point, you can make discoveries really quickly. We started with a set of lung cancer patients' data, early stage lung cancer patients' data, and within a month had these types of models that I'm describing, and we're able to screen billions of drug-like small molecules, including drugs that were already clinically being tested. So one of the things you were talking about with this process with cores was, how do you get to proof of concept quickly? Well, if you're not so invested in one target, if you're more invested in validating an end-to-end approach, which is what we've done in lung cancer, for example, then you could take drugs that are already safe for people because you're not so wedded to whether that drug is your drug or someone else's drug. And you can prove that this entire approach works. You can find molecules that will move the needle for this set of patients. And you can actually test that very quickly in vitro, in vivo, in mouse models and so on. And then you can even go do the clinical trial if you have the funding to do it. We were just talking about that with one of our collaborators, Dr. Charles Powell at Mount Sinai, which is that, hey, we found drugs that look interesting. They are innovator drugs from some other company, but because they've been through the clinic, you could get right into a phase 1-2 clinical trial. And we just started this work months ago. That's really fast to be able to get that far that quickly. And so I think that's what's possible now with these types of computational approaches.
So you're talking about basically AI-driven screening and how that speeds up the treatment, right? Or research and development. And to take for years to months is kind of the example you just gave us there. Can you tell us a little bit about maybe the origin of Archetype Therapeutics and how you guys came up with these models, and what do you really think this type of screening, I know you gave the example already of years to months, but how is this going to play out over time? And by the way, just throw it in there, I love the drug repurposing concept, because I think that there are lots of molecules that are out there, lots and lots of them, and we've seen a number of efforts to do it, that might do all sorts of things that, you know, people have kind of thought about, but just haven't been kind of the central way that they got to market yet. And there's just this enormous opportunity across that space. So maybe you tell us a little bit about the screening and how you got there, and then a little bit more about what types of molecules you think are kind of best to use that approach for.
Yeah, sure. So as you know, we talked a little bit about my background, and my co-founder in this company, Paul McDonough, has a kind of parallel background, but he's the hardcore scientist and the guy that developed the platform. So he was working on the completion of the Human Genome Project 25 years ago. He was early at Rosetta Informatics, where they were doing the gene expression work in drug discovery a very long time ago, over 2 decades ago, working with pioneers like Eric Schott and others. And so he's cross-trained. So he did that type of work, but then he also did work in patient care associated applications. So there was work done at Rady Children's Hospital with Dr. Kingsmore. Paul was part of that team, and they profiled NICU babies. So they did clinical genomic characterization of NICU babies who had rare diseases, who were on the verge of death, essentially, and built this whole system to be able to take a heel prick from the baby, get blood from that, do a whole genome overnight, reason over that data and the clinical data, and identify what was wrong with the child, and then identify whether there was a drug already available. And sometimes it would be something really simple, like a vitamin B12 deficiency. They saved a bunch of kids' lives. They won a Guinness World Record for that. So part of understanding how we came up with this approach that I'm going to describe is we have this kind of under the radar data scientist rockstar in Paul that is cross-trained for 25 years across all of these different genomic and clinical data applications, both patient-facing as well as drug development-facing. And so that's where his ideation really came from. He started working on this idea about 2.5, coming up on 3 years ago, which was, given all these new AI tools, including some of the generative AI tools, can we, instead of being focused on the old school approach of discovering a target and then trying to find a molecule that hits that target, which is still a very valid way to do drug discovery, but you might be missing a lot of the biological picture when you just focus and narrow in on just one target. His concept was, well, there are these, what we just described a couple minutes ago, these human clinical genomic classifiers. What I mean by that, for those in the audience that may not be aware with all the jargon, is this concept of something like a Cologuard. So most people know what that is. That's the poop in a box test. You essentially want to get screened for the possibility of colon cancer. You provide this stool sample. They do expression profiling. What that really means is measuring the activity of your genes. And depending on the activity of your genes from that sample, you may be more likely to have or develop colon cancer. Well, those types of tests exist for all manner of diseases, including many cancers. And what they do is they predict the likelihood that you're going to have the disease, or the likelihood that if you have the disease already, that you're going to have a bad or relatively good outcome in terms of how long you live or whether your cancer spreads and so on. So that's what we mean by clinicogenomic classifiers. And those are great to predict what will happen. But what happens if the prediction for you is that you're not going to have a good outcome? What if you're an early stage lung cancer patient and the prediction is, actually, even though we caught this early, you're not going to live very long, or your cancer is going to be very aggressive and it's going to spread? What do we do for you? So great to have a prediction that helps doctors and patients think about how aggressive to be with treatment. But what if there isn't a treatment for those patients? So that was Paul's kind of original idea, was what do we do for those people? And then he grabbed all the latest AI tools and said, well, what if we could predict for molecules that perhaps haven't been tested for this disease? What if we could generate the effect that that molecule would have on the biology of that disease? So how is it going to affect the expression of all the different genes that matter for the disease at once? Not just one particular gene, so one target, but rather a dozen or several dozen genes at once. If we could do that, then we could put that together with that understanding that we have from the human patient data and say, okay, let's take a molecule, or let's take 1 billion molecules that look like drugs. People have built libraries of molecules that look like drugs, but you can't test all of them because you can't test 1 billion drugs. There's not enough reagents and solvents in the universe to do that. So what we can do now is we can do that virtually. We can take 1 billion molecules in a day at this point, screen them all and say, what are they going to do to the biology of prostate cancer or colon cancer or lung cancer? And then project that information into these patient cohorts and say, if I could make these changes in these actual people, I would increase their lifespan by 2 years, for example. So that's how we do this. We've merged these two. One that's kind of an older capability that's been around for 20 years, which is these classifiers in Cologuard and these other types of tests. And then these generative AI capabilities that are relatively new. We couldn't have done this 5 years ago, but these types of algorithms allow us to understand what billions of molecules, things that look like real drugs, but we can't test them all physically. What are the ones that are most likely to make changes to cancer biology that will matter for these patients and move their outcomes in the right direction? So that's this massive screening that we talk about. That's what we mean by that. And so we've screened billions of molecules in prostate cancer, in lung cancer, and we don't get some kind of esoteric readout. We get a readout as to what will that do for a set of patients that we know have unmet medical need. And so once we have those answers, what are we doing? Well, we're saving all that time and expense on testing physically molecules that are unlikely to actually make a difference for patients. And so that's why when I mentioned that example earlier, why did it take only months to identify both drugs that people already know about as well as novel molecules that can work for this specific type of early stage lung cancer? The way we were able to do that is through that massive screening process that I described that happens virtually first, saves all the money and time, so that we can focus in the time and effort and the use of the expert biologists and clinicians on the much smaller set of de-risked molecules that are likely to actually work for these patients.
So when I think about what you're saying, and I just think about my own experience, I started in computational chemistry and genomics, right? We were building libraries of, what are all the molecules we could build if we have the following reagents and the following robot, and let's just model those out. And then let's look at what we could make, and was it drug-like or not? We were trying to use certain characteristics that describe, this looks like a drug or has the characteristics. So it was the Lipinski rule of 5 at the time was the big thing. And could we make those molecules? And then we were actually working with pharma companies that had in their mind the idea that, we're just going to make all the drugs and patent them all. We'll just own all the drugs before they even exist and nobody's ever tested them.
Right.
So there's a lot of effort around high throughput screening. So it sounds like we're taking the high throughput screening. There's a systems biology component that you talked about in my mind, which has always been, I think, kind of a major issue. And it sounds like you're saying the AI tools are there. And the issue that I talk about is, you think about what we know about biology, I always feel like it's way less than we think it should be. We actually understand biology much less than the actual, the body is really complicated. These systems are incredibly complex. But it sounds like now with the tools that we have in place, we're able to either model the systems biology, right? Or maybe it's something that I think you might have said in here, which is understand from the real world data, from the actual patients, the patient centricity of what, even if we don't understand exactly what happens, what do we expect to happen when certain things change? And you're kind of meshing that all together with the result of taking the risk out of, like, when you actually go and do something for real, then it's much more likely to be successful. Am I tracking this?
Yeah, I think so. I mean, when a lot of the work back when I first met you and I was working with the folks at ATIA back when the company was called GNS Healthcare, and Paul McDonough, my co-founder, was also there, and we were early co-inventors of the technology there, still working with those folks and love what they're doing. And we were very hyper-focused on kind of the detailed causal biology that we could learn from all the omic data. This approach is a little bit different in the sense that, exactly as you just said, we may not understand all of the detailed mechanism underneath the hood, but if we understand what changes are associated with better outcomes for those patients, and if we can in fact induce those patterns in these people, the hypothesis, this completely different approach that we've adopted, is that we will enrich our hits. It won't be perfect, but we'll enrich our hits and we'll massively speed this up. So scale, speed, and relevance. Optimizing for all of those things at the same time. You're not going to be perfect at all of those, but we think that we've struck this right mix, and we've kind of proven that by going through in vitro and now in vivo models that you would put regular drugs, going through a regular drug discovery process through, and finding validation at each of those steps very quickly. So and I would also say that there are a lot of AI drug discovery companies now tackling different aspects of whether it's target discovery, or I mentioned there are companies, even old school companies, that have been at it for dozens of years doing structural biology or AI characterization of different types of targets, or the ADMET and toxicity type aspects. So there's so many companies out there. I don't think we have all the answers, but we think that by trying to start with the patients and finding out what will move the needle, move them towards states associated with wellness, that we might be able to short circuit or get around some of the kind of length that you alluded to earlier in the conversation. And so far, we have to bring some of these discoveries we've made into human clinical testing, but we're almost on the verge of that now, having gone through in vitro and in vivo studies that validate that the type of approach that we just talked about actually works, that you can start with 1 billion molecules that look like drugs, identify just a few that look good, and actually prove out through the normal processes that every drug gets put through the same paces and meet those standards. So that's what's exciting, is that this is now possible. And this is really hot off the press over the last few weeks that we've gotten some of the in vivo data, which is really kind of the metric that people look at to say, hey, this is cool. Cancer's been cured in Petri dishes millions, maybe billions of times. So the bar is a little higher than that, but we're starting to show that this is possible.
This is amazing, Tom. I'm so excited to see it actually coming to fruition. Your point about it being probably the last few years, I really do feel like the last 2 or 3 years, Gen AI, and then beyond Gen AI, just the amount of data and understanding that we have in this space, like these computational models are finally ready to pay off. I'm really looking forward to hearing in the near future, hopefully, about the kind of results as you start to move these things even further forward. Let's talk a little bit about like the future. Like if we got to here, and it only took us 20 years, right? Actually took even longer than that in the AI space, but it feels like things are moving really, really fast, right? So if we think about, maybe 5 years from now or 10 years from now, what do you think is going to be happening? Like, what is this current generation or revolution in technology? Like, what's going to happen next? And I'd love to hear your thoughts. I have a few myself because it's come fast.
Yeah, it has. That whole question around, think about the singularity and the elbow of the curve, and where are you in that elbow? And, once you enter the curve, you feel like you're in a straight line, but actually things are going to move very differently. It's kind of felt like that a little bit in the past, but now, of course, with the benefit of a few more years of perspective, it just feels like the speed at which the next set of algorithms, the next set of improvements is arriving is much faster. That what we worked on for 5 years and said, hey, this is a pretty cool improvement in network biology, is now, well, in 3 months, someone's come up with a better version of an LLM or some other kind of AI-based tool that they're deploying against these problems. So I think that's one thing, is that the speed, the acceleration is increasing. That seems evident just last year, how quickly things moved last year. So one other aspect that I think will be interesting in the next 2 or 3 years is how do all these different incredibly exciting innovations converge? So I mentioned things around target discovery, things around what we're doing in high throughput screening, things around ADMET and the other properties of the molecule that make a molecule actually practical, things about how you actually deliver the molecule to the right cell or cell type. That's really exciting. And there's been a lot of innovation around that as well. So I think part of what I think is going to happen in the next couple of years, or at least needs to happen in the next couple of years to make this all really impactful, is who's going to aggregate all these different types of technologies to solve these problems. Now, obviously, the big pharma companies are trying to do that, and they have the resources to do that. But I think it will also fall increasingly on smaller, more nimble, innovative companies to partner up with each other. And we're trying to do quite a bit of that at Archetype. We have limited bandwidth as a small company, but we're watching these other companies that are bringing other parts of the puzzle from an algorithmic perspective. And then again, talking about your company, XCures, and other companies, the data piece. That piece also is accelerating. How well can we characterize what's happening in disease? So I've played in gene expression for a long time, but that's not the be-all, end-all. There are all these other omic modalities. There's a spatial aspect to everything that happens in biology, and there's a temporal aspect to everything that happens in biology. And with the cost of all that profiling going down, access to all that data and the ability to do these iterative loops of generate a bunch of data, learn something, test it, take that information from the test, loop it back in, learn a new model, make a new prediction, and so on. These are the types of things that I think will and need to happen over the next 2-3 years to see a real impact. I mean, right now there's a lot of hype around AI drug discovery, and then some of the naysayers will say, well, how many truly AI discovered drugs are there on the market or even in phase 3 clinical trials? And that is still pretty limited. I think everything that I just described coming together, both with smaller companies making a purposeful effort to connect the dots, and then some of the bigger companies getting access to the pieces, that will ultimately drive a situation where I think you'll just have two things. You'll have much more specific and personalized therapies because it'll be possible to characterize patient groups in a much more fine-grained way. And then what we've been looking for, which is both an increased speed and a decreased failure rate in late-stage clinical trials. Those are the things that haven't happened yet, despite all the excitement and the hype, and that I think can happen if these pieces get integrated over the next few years.
Yeah, I totally agree with you with the hype. I feel like every time and I'm a technologist, right? Have been my whole life. And so I'm really excited about technology. And then I always get called out with like, yeah, but we talked about that for a long time and it didn't actually change anything. And we spent a lot of money on it, right? Developing it and a lot of time and effort. And maybe we should have just done it. Like that's kind of the laggard thinking, right? Or the naysayers, which is just like, yeah, you did a lot of stuff. It was pretty cool, but it didn't do anything. So why did we invest in that? I think your point about convergence is one that resonates a lot with me. And as I think about convergence and AI tools, and I'll talk a little bit, I've been doing a lot of work in healthcare, right? In healthcare processing, like just what happens to patients when they try to go to the healthcare system and they don't have their data with them. So how do we get the data just to facilitate the, basic things like going to get a surgery or going to see your doctor or getting treatment for your disease where you have to share information from all sorts of different locations or different provider and healthcare systems. And that data is part of what you were talking about, that then becomes kind of core research enablement later. But it often just starts with processes that exist today. I think of things like, you're a new patient at a hospital, they have to get all your data and organize it. Are they getting it on a fax machine? Is someone typing it in? What's the AI to stop all that human cost and labor? And when I take those concepts and apply it and start thinking about what I know about drug discovery, right, and development, and that space, to your point, it's kind of a convergence, right? There's the gene expression, There's all the omics, the spatial components you talked about, the overtime exposure. There's patient, profiling, demographics, history, exposure to both environmental, socioeconomic influence, right? Like we're starting to understand that the world is really complicated. And then there's also all of the stuff I think related to, I think about the hardcore kind of drug delivery. So like material science, we're seeing like ultrasound therapies, right? Coming together, like all of these different technologies are coming together. So it's almost like there's all these amazing tools being generated really, really quickly. And I feel like the AI is making them accessible. They used to be only accessible to specialists in a very, discrete, high cost environment. But if we've now generalized those and democratized access to, let's say, the best thinking in material science, right, that's going to help us with some drug delivery thing, and the best thinking, it starts to put the challenge as not, do I have the right tools, right? Or can I get access to the right tools, but can I put together the right pipelines, right? Going back to your concept of like, how do we, what is our objective? What's the goal we're trying to get to, right? What are the traditional ways we go for it? What are the bottlenecks along that chain? And then how are we going to get all of this technology, right? And use the right one, right? So it's a question about selection and application. And so, as I think about that, I get really in my heart focused on like, we have all this stuff, like what should we be trying to solve, right? Like what is the fundamental things that are going to have the greatest impact? And I think about impact in the context of kind of really two things. I think of social benefit, right? Personal benefit, like healthcare benefit. And then I think about economics because, we live in a world where economic benefit is due. And I think there's this like intersection of those two, which is kind of like the magic crossover where not only we do good for people, we help cure diseases, right? We develop new drugs, but we actually provide economic value to all of the participants. And that's been really elusive, right, for a long time, right? If you just think about, great, we have a new drug, well, who's going to pay for it, right? And who gets access to it? And we have to find a way to balance those two things. And so how do you think about like the drug discovery and the cost and then bringing these things to market? Do you think there's an opportunity here that we can actually do these things more effectively than in the past?
Yeah, I mean, look, first, the why is super important. And so going back to what you were saying earlier about your work on the patient side, look, I wish XCures had been around 15 years ago in the form that it's in now. I had two friends that I lost, one in her 30s and one in her 40s to cancer. And I'm not that old myself. I'm not 50 yet. So, and so I was in this space when both of these friends of mine were diagnosed and so on. One with colorectal cancer and one with a rare appendiceal cancer. And so, first, they needed some of the stuff that you described. They needed all of their data in one place. I was helping them kind of just grab manual charts together. We were trying to find clinical trials. Now there are all these great companies, XCures for the patient data, companies like Massive Bio to find a clinical trial for them. All that stuff didn't exist 15 years ago, and it does now, thanks to the work that you and these other folks are doing. So that's one aspect of it, is we can actually do some of that stuff now. Then you got to connect it to, okay, well, as I was saying earlier, do we have the drug for them? Even though we now have a lot of the infrastructure we didn't have before, we may not still have the drug. And to your point, it may not be financially feasible to provide that drug to that person, even if we can identify them, get them, find where they are in which patient cohort, find the drug that's right for them, and so on. Either the drug doesn't exist or there's an economic impediment to that. Why are there these impediments? We're not going to solve all these problems, but some of it is just this massive inefficiency that we talked about. 10 plus years, $1 to $2 billion to produce that one drug. A lot of that is because a lot of the processes that occur are physical along the way. So the screening, if you're doing high throughput screening, that's physical. You got to test 10,000 or 50,000 molecules at the very beginning of that process. And most of those aren't going to work. You're not casting a wide enough net. You might not be finding the one that does work, and yet you're still spending a bunch of money, for example. And then doing all of that testing in animal models without having ever touched any human data, and then going into a human trial and expecting and hoping that the drug is going to work. So a lot of these things are now addressable and are being addressed by the types of things that we talked about earlier in the conversation. So I do feel optimism that we can squeeze a lot of cost out of it. Now, We don't control all of that. How does that result in better pricing for the drugs and so on? Some of that is out of our hands as a little company, but we can enable that possibility with some of the things that we talked about in terms of making the process faster, more efficient, less late stage failures. That's one of the biggest drivers of cost, because those trials are really expensive, and often the de-risking that can be done through some of these computational approaches has not been done. And then the drug either makes it or it doesn't, and somebody's lost $100 million in late stage clinical development, and we don't have a drug for those patients. So I think that economic opportunity is there. Whether or not the system as a whole, makes the right adjustments to pricing to be able to deal with that aspect of access that you alluded to, I'm not sure. But I think we can make these things less expensive to develop through all of the technologies that we've talked about.
Tom, thank you. I have one last question for you. If there was one thing that you and I or others, or that you know of, that people aren't working on, but that you wish that somebody was working on, right? That you'd like someone else, what would that thing in AI be today? Like, what is it that's out there that's missing that somebody should try to do?
So I'll talk about something that I wish could be done on a grander scale. I do know some folks that are doing it on a smaller scale. So I'll mention what they're doing and then why it should be happening in a much broader scale across all data. This issue of, and this has hounded me from my days at ATIA that we were talking about going back 15 plus years, again, at another then small company fighting this battle where we had these great analytic tools. It was access to the right data to solve these problems. A lot of people have the right algorithms and don't have access to the right data. And so there are some companies that have emerged that are doing a really bang up job of grabbing and sucking up and hoovering from all the corners of the world, all the relevant data of a particular type. So for example, my friend Bobby Sieber at Panicent is doing this with single cell data. So they grabbed all largest single cell data repository across human, non-human cell types. That's really important because ultimately, if you're trying to treat a disease, you need to know what cell or cell types you're actually treating the disease in. And often when in cancer, for example, we get these bulk tissue samples, and they're a mix of normal cancer cells, immune cells, stromal cells, so on and so forth. So being able to deconvolute all of that, first physically having the data, and then being able to use AI to deconvolute, companies like that company are solving that. But that's single cell data. What about all the other different types of data modalities out there? I think that the AI tool explosion will continue, and the ability to readily access the right pertinent data to solve the problem that you're trying to solve is going to increasingly be a question of, can you get the right data in your hands, rather than does an AI tool or set of tools exist to do it? So there's a lot of progress in that. We've talked about what your company does and other companies do toward that, but I still think there's a meta layer of connection of all of that needs to happen. And when that happens, the ability to take what 1,000 companies are working hard on from an AI perspective and fully maximize it becomes possible.
It's true. One of the founding principles when we started looking at cancer and X cures, right, was the difference between collaboration, right, and independent discovery. So if people take a strategy of collaboration, if you share information and learning, the participants in any system actually progress faster than if they don't collaborate. But that's kind of anti-sensical in some ways, many times when you're thinking about companies and developing IP value and all the rest of it. And so understanding how collaboration and coordination, right, which is an even deeper form, right, of that, which is how do I do one thing, right? I have my time and energy, and you do, the opposite thing with your time and energy, but we agree to share what we did so that we both learn, right, from each other continuously. Like, I think that's a huge, huge opportunity. Well, Tom, thank you so much for coming on AI and healthcare. This has been an amazing conversation. I always love talking to you. It's fantastic.
Likewise.
Thank you so much. I'm really looking forward to Archetype, Therapeutics and what you guys are doing and kind of the next chapter that's unfolding here.
Thanks so much, Mika. Great chatting with you.